Welcome to Better You. In this video, I'm going to give a quick review of ANF, anti-nutritional factors, the classification, and certain examples that are important for your examination. So first, let us look at what is the definition of anti-nutritional factor. So they are substances which are either by themselves or through their metabolic products interfere with food utilization and affect health and production of the animal. So as the name suggests, they are the nutritional factors which affect the nutrition. Okay, so anti-nutritional factors. They are usually secondary metabolites found in virtually all the plants to some degree, but they are more common in tropical forages and they have a defensive role in the plants. So the important point here being they are present, or we can say they are more common in tropical forages. They are also referred to as toxic factors. Okay, and they are the substances which, by themselves or by their metabolic product, interfere with the nutrition. Okay, so they are classified on the basis of their chemical structure into four groups: Group 1, Group 2, Group 3, and Group 4. Group 1 is ANF that are proteinaceous in nature. So they are proteins, for example, the enzymes like protease inhibitor and hemagglutinin proteins, phytohemagglutinins. Then the second group is glycosides, so they are chemically glycosides. Okay, examples are saponin, cyanogens, and goitrogens. Group 3 is phenols like gossipol and tannins. And group 4 is the miscellaneous category, which includes anti-minerals and anti-vitamins. Okay, so this is the classification on the basis of chemical structure. In this, you just need to remember these categories. Okay, now the second basis of classification, that is on the basis of nutrition that they affect. This is very much important from an LAQ point of view. In exam, they ask long answer type question from this. Okay, so first we'll look at the category of substances interfering with the digestion and metabolism of protein. So they're anti-proteins. They include protease inhibitors, so the enzymes, or we can say the substances which inhibit the protein digesting enzymes like trypsin and chymotrypsin. So we have Kunitz antitrypsin and Baumenberg trypsin and Baumenberg chymotrypsin inhibitor in soybean. This is a very important example. Kunitz antitrypsin and Baumenberg trypsin chymotrypsin inhibitor in soybean. Then we have phytohemagglutinins. So they are hemagglutinins. They are proteins which agglutinate, or we can say which clump the RBC. Okay, and they are of plant origin. That is why they are called phytohemagglutinin. Okay, they are also known as lectin. Important examples include robin and ricin. Ricin is present in castor bean. Then we have saponins. Saponin, as the name suggests, sapo means soap. So they are the substances which form soap and they lead to bloat in ruminants. Okay, and they are present in lucerne, white clover, red clover, and soybean. Again, very important example here. Ricin is also important for phytohemagglutinins. Then tannins, they are the substances which bind to the proteins and precipitate the proteins. Okay, so basically they lead to decreased absorption of the proteins. And there are two types: hydrolyzable tannins and condensed tannins. Condensed tannins are not easily hydrolyzed by the enzymes of the body, so they are more effective at decreasing the protein absorption. Examples include amygdalin in bitter almond. Tannins are a very important group of compounds because when they are present in the feed unintentionally in higher doses, they will act as anti-proteins, but you can utilize them intentionally to form bypass proteins, proteins which are not digested in the rumen. Why? Because as it suggests that they bind and precipitate the protein. So the protein is unavailable for the digestion by the microbes, but we will use such tannins which are not so much condensed, right? We will use this type of tannins, okay, and they will not be degraded in the rumen, but when they enter into the intestine, they are able to be digested by the enzymes. So the protein has bypassed the rumen. Thus, the tannins are used intentionally in the production of bypass proteins, which are used in feeding of high yielder animals, especially high yielder dairy animals. Okay, but if it is unintentional, the tannins are anti-nutritional factor. Then we have substances which reduce the solubility, and utilization of minerals. So first we had anti-proteins, which included protease inhibitors, phytohemagglutinins, saponins, and tannins. The second category is anti-minerals. So they include phytic acid, oxalic acid, glycosinolate, and thioglycoside, and gossipol. So phytic acid form complexes with all these minerals, but the most important one is phosphorus. Okay, then oxalic acid, again it forms bonds with multiple ions, but the main one is calcium for exam. Then glycosinolate and thioglucosides, they are present in the members of the Brassica genus. Okay. Second thing, that they prevent the utilization of iodine by the thyroid gland, so they act as goiterogens. They lead to goiter. And gossipol, it is a polyphenolic compound present in the cotton seed cake, and it binds with iron. That is why the use of cotton seed cake should be very cautious. Okay, if you are using the cotton seed cake in animal feed, then you should know that it may contain gossipol. Okay, and what will gossipol do? It will cause iron deficiency because it binds with the iron, precipitates it, and decrease the absorption. Gossipol toxicity is more common in monogastric animals, and it causes egg yolk discoloration and yolk mottling. This may be asked in the exam. They can ask you this point. Okay, then we have antivitamins. So, as the name suggests, they are anti-nutritional factor which act against the vitamins. So, they include antivitamin D in soybean, antivitamin E in kidney beans, antivitamin K dicumarol that is present in sweet clover, antivitamin B6 present in linseed. Other than that, we have antivitamin niacin which is present in wheat bran, and antithiamine present in bracken fern. Out of these, the bracken fern is important, kidney beans is important, and dicumarol in sweet clover is important, and soybean. Okay. Then we have substances interfering with the metabolism of carbohydrates. So they are ANF acting against the carbohydrates. They include amylase inhibitor, which is present in oats and rye, and phenolic compounds, and flatulence factor. Flatulence factor cause increase in the gas production in the rumen, thus they lead to flatulence. Then we have anti-hormones. So they are the compounds which act against the hormones like mimosin. It is degraded to dihydroxypyridone in the rumen, and it reaches the thyroid gland where it inhibits the synthesis of thyroid. Okay, and it is present in subabule. Then we have cyanogens. These are the compounds which release the HCN. Okay, and ruminants are more susceptible to cyanogenic plant toxicity. HCN is present in rubber seed cake, linamarin present in linseed, and durin is present in Sudan grass and sorghum. So durin is the most important example of cyanogens. Then we have goitrogens, as we have discussed. Glycosinolates and thioglycoside, they interfere with the absorption of the iodine, while mimosin interferes with the utilization of the iodine to form the thyroid hormone. Okay, and both of them lead to deficiency in iodine, basically, or we can say deficiency in the thyroid, and they lead to goiter. At last, we have the alkaloids, which include atropine from Atropa belladonna, which is also called deadly nightshade. We have strychnine from the seeds of Nux vomica, and solanine from potato. Out of these, atropine is the most important one. Okay, so this is all about the examples of the anti-nutritional factor. Now let us look at some examples which are important from an exam point of view. So we have soya bean. It is often asked in match the column or some other objective type question, which are the anti-nutritional factors present in soybean. So we have trypsin inhibitor, right? Kunitz trypsin inhibitor and Baumenberg trypsin chymotrypsin inhibitor, they're important. Other than that, antivitamin D and A are important. In groundnut, they will ask about aflatoxin. In mustard and rapeseed, they will ask about thioglycoside and glucosinolate. In cottonseed, they will ask about the gossipol. Castor seed has ricinin. Linseed has linamarin. Neem, nimbin. Karanjin. Maua cake has maurin. Rubber seed has HCN. Barley has beta-glucan, fish meal has gyrosin, bracken fern has dicumarol, kidney bean has antivitamin E, and jowar and Sudan grass has durin. Okay, so these are the important examples that you should know about anti-nutrition factors and the feed ingredients which contain them. Then one LAQ is asked on various methods of removal of anti-nutritional factor from the animal feed. Okay, so suppose you are using something which may have anti-nutritional factor and you want to decrease the level. So what are the various methods which you can utilize to remove the ANF from the animal feed? Okay, so we have autoclaving. So if you have studied microbiology, you know what autoclaving means. It is a moist heat. Okay, basically we use steam along with pressure, and it is useful for detoxifying toxic factors in the feed like ricinin in castor bean meal. This is important. Then it also has the benefit of killing any harmful microbe in the feed. Okay, but the one downside is that it can degrade the temperature sensitive nutrients like vitamin C or some other vitamins. Then we have roasting. It is a form of dry heat. Okay, the temperature is important, 149 or 148 degrees Celsius. form of dry heat that can inactivate the enzymes. Okay, we can use roasting for soybean to inhibit the trypsin inhibitors. Then we have soaking. So for soaking, we soak the grains for 12 to 24 hours. Okay, the roughage stuff, the dry roughage can also be soaked, but here the soaking is mainly used for grains. Okay, we have to soak for 12 to 24 hours. The time is important. 12 to 24 hours before feeding. Okay, and it will remove factors that are soluble in water like tannins. Okay, and that is why we say soak the almonds in the water 12 to 24 hours before consuming them. Right? Why? Because almond, as I've said, it has amygdalin, which is a tannin. And when you put almond in the water after one night, you can see that the water turns brown. Why? Because the tannins have been released into the water. Okay, then steaming. Steaming uses moist heat, basically steam. It acts like autoclaving. The only thing that is different is pressure. In autoclaving, we have pressure. In steaming, we don't use pressure. Okay, the temperature is 100 degrees Celsius for 8 to 20 minutes. Then blanching. In this method, the feed is first scalded in boiling water. So we utilize very hot water to first give heat treatment, and after that, we plunge it into ice water to give it cold treatment. So basically, we are giving heat shock. Okay, it stops the action of enzymes and inhibit toxic factors like amylase inhibitor and trypsin inhibitors. Then we have extrusion. It is a process of cooking in which feed are also expanded by the application of adequate pressure. Okay, and it controls the growth of harmful microbes and remove toxic factors. So this extrusion, the name is mostly important. Then pressure cooking and normal cooking. Pressure cooking is mainly used for detoxifying legumes like right kidney beans. So these are the various processes which you can utilize for the removal of ANF, anti-nutritional factor. And this is it for the topic anti-nutritional factor in animal nutrition. If you found this video useful, then hit the like button, share this video with your friends, and if you haven't, then subscribe to my channel. Thank you.